Good morning and good afternoon to all of you for joining our live webinar today, Functional Characterization and Substrate Binding Kinetics of the Sodium Cotransporters SGLT1 and GAT1. Today's webinar marks the second talk of our Research Insights Transporter webinar series for 2022. My name is Jason Villagomez, Marketing Manager at Nanion Technologies, and will be your moderator for today's event. I would like to welcome everyone on behalf of the whole Surfer team. Thank you for joining us today for the second session of our Transporter webinar series. In particular, a welcome to Andre and Rocco, our speakers for today. It is a pleasure having you today and for sharing your exciting research with us. As mentioned, this is the second Transporter webinar in 2022. The next session will be about charge translocation reveals new insights into the transport mechanism of human antiporters, and that will happen on November 17th at 4 p.m. And we will send out invites for that shortly after this meeting. In addition, we welcome you to access our on-demand Transporter webinar series from last year. They're available on our website and also our YouTube playlist. We will share the playlist in the chat. And finally, if you have further interest in our technology, don't forget to join us for our user meeting in Munich. That's happening on October 19th through 20th. We're having a dedicated session on the Surfer technology. Once again, as presenters, we have Andre Bazzone, Senior Scientist of the Surfer team, and PhD student Rocco Zerlotti. Andre and Rocco will present on studies on the sugar transporter SGLT1 and the neurotransmitter transporter GAT1, revealed detailed kinetic information from substrate-induced electrogenic pre-steady state reactions. But before we get started, some due diligence. Upon completion of the presentation, we'll hold a live Q&A session with the presenters, and as such, welcome you to submit your questions at any point through the course of the presentation. If you happen to experience any technical difficulties at any time, please inform us via the chat box. In addition, today's presentation will be available on demand shortly upon completion of the event, inclusive of a Q&A session. I will now turn over the presentation to Andre for the start.
Thank you, Jason. So I'm actually want to welcome everybody as well, and also a welcome from the whole Surfer team again. I'm going to talk about the sodium-coupled sugar transporter SGLT1. And this is a kind of unusual webinar. Usually customers present their work here, but Nanion actually not only develops and produces devices, we also are involved in several research projects and have lots of different internal projects running. So I'm talking about this from my part, from my point of view, and after myself, Rocco, a PhD student, will talk about his project on the sodium-coupled GABA transporter. So I can actually take the laser pointer. Before I actually dig into the biology of SGLT1, I want to highlight a few things regarding the Surfer technology or SSM-based electrophysiology in general. There are a few principles behind this instrument which differ from other conventional technologies like patch clamp. First of all, the Surfer reflects a robotic pipetting system, so it actually injects solutions with the substrate or ligand of your transporter of interest into a sensor well containing your transporter of interest. And this substrate gradient triggers your transporter activity. And this happens at 0 millivolts. This is in contrast to, for example, voltage clamp, where you actually do voltage steps to trigger the transporter activity or the ion channel conductivity. The sensor works as a capacitor. So you have lots of vesicles attached to the solid supported membrane. When you actually inject your substrate to the solution, it triggers transport activity, and this charges your vesicular membrane, which itself then leads to the charging of the measurement electrode below. And this charging current is what you are measuring. This also means that you don't observe real steady state currents because you lose driving force over the course of the measurement. You have a concentration gradient that leads to the generation of membrane voltage, which at a few hundred milliseconds upon transporter activation basically equals the driving force of the substrate gradient, and therefore transport is completely blocked. So you will see a steady decay of your transport current, which is measured. And usually if you observe transport, the peak reflects your steady state transport activity. The sensor is sensitive enough to also measure pre-steady state currents up on binding of the substrate, which might be the case for some transporters, as you will see later. And here the decay of the current is much faster compared to the decay resulting from transport. So this is something just as a reminder. Now coming to our results for the sodium coupled sugar transporter SGLT1. Actually, most of the things I'm going to present are unpublished. Here's just the first slide, what has been published already, and is the very base of the things which are following. I will just quickly guide you through this. On this left-hand graph, you can see currents induced by different substrate jumps in presence or absence of co-substrate recorded on SGLT1 vesicles. In the center, you will see the same currents, the same experiments basically carried out on vesicles not containing SGLT1. This is a negative control. And on the right-hand side, you will see basically the subtraction of the negative control from the SGLT vesicles. So this is your pure SGLT1 current. Let's have a look first at the dark blue trace. You can see a schematic of the experiment here. This is the result of a glucose concentration jump in presence of sodium, in presence of co-substrate. And you can see that you induce a fast current here and then a slow decay, slow current decay afterwards. So it's a biphasic current, and we attributed that slow decay to the actual transport current, reflecting sodium coupled sugar transport, and the peak current, the fast decay, we believe represents sugar-induced conformational transition, likely induced fit of the sugar molecule. So this is happening in presence of sodium. We repeated the same sugar concentration jump in absence of sodium. This is the light blue trace. You can see that the transport current phase is absent. So you don't have any sodium coupled transport because there is no sodium, but you still see the fast current decay reflecting the sugar binding. This is actually here represented. So we replaced sodium with potassium in these experiments. Then we did also sodium concentration jumps in presence of glucose. And you can see again the transport current phase because both substrates are available, plus a somewhat smaller pre-steady state current phase. And finally, we also did a sodium jump in absence of sugar. And here, if you see the subtraction of the negative control, it's basically a flat baseline. So you don't see any electrogenic event up on sodium binding to the transporter. So this is a very basic. After that, we actually thought about, hey, why not using the availability of the sugar binding readout to measure sugar binding kinetics of SGLT1. So we did a concentration sequence of sugar, of glucose, And here are the currents for the different glucose concentrations. Again, we see pre-steady state currents and the slow decaying transport phase. What can be done is actually using the slow decay and reconstructing the real steady state current using circuit analysis. This is actually a MathCAD script you can apply, and then you will get from that decay just a steady state current, and you can nicely fit the concentration dependence of the steady state individually from the pre-steady state, which we did. So we used the steady state current concentration sequence to conclude a Km value for the glucose, which is 1.9 millimolar, and matching nicely with literature data at 0 millivolt conditions. And we also took the pre-steady state peak, and we believe that the EC50 from the hyperbolic fit represents the affinity for the sugar to the sodium bound carrier, because the pre-steady state is a direct consequence of sugar binding. So what else can we do? Actually, if we assume that after sugar binding with the KD, there is the electrogenic induced fit happening, which is recorded in during the surfer experiments, we can actually measure the rate constants of that induced fit. And for that, we used this model equation, and we just need to detect the K-ops, the rate constant of the pre-steady state. We did that by just fitting a bi-exponential decay to the concentration-dependent transient currents. And as you can see, the slow current phase is not so interested. It's not so interesting here because it's reflecting the transport phase. We just look at the fast current phase, and the fast current phase actually changes with sugar concentration. So we can use this equation to do the fit, and we can get a KD value again, and also the on and off rate constants for the induced fit. I also just, out of curiosity, plotted the concentration dependence of the second tau, the decay reflecting the actual transport phase. And here you don't see any concentration dependence. So now we basically measured the kinetics for sugar binding to the sodium-bound carrier. But we also saw that sugar is able to bind to the empty carrier in absence of sodium. So we basically repeated the concentration sequence for the sugar, for the glucose in absence of sodium. And as a reminder here, the transport phase is completely missing. Makes sense, there's no sodium available. We plotted the charge, the integral of the currents, to obtain affinity for D-glucose to the empty carrier, which is like 200 millimolar. So much higher, 2 orders of magnitude higher compared to the Km value, which is very interesting. I will come to that later in more detail. And also we found that the decay time, it's mono-exponential, of course, because there's no transport phase, and the rate is not dependent from sugar concentration anymore, as you can see here also for the K-ops plot. So now we know KD affinity for the sugar is way different when sugar binds to the sodium bound carrier or to the empty carrier. So we became interested in how does the affinity actually changes when you have different sodium concentrations. So we performed 30 different experiments using 30 different conditions on the same sensor, pushing the limits of throughput of the surfer N1. So what you can see here is in one plane a certain sodium concentration which has been used, and within the same plane you have different sugar concentrations highlighted here. So this all has been recorded on the same sensor. We again reconstructed the steady state current. So this is basically to distinguish pre-steady state from steady state. We then did, again, the hyperbolic fits to basically conclude Km and Kd values for the sugar, shown here, in presence of different sodium concentrations. And since we also have different sodium concentrations and sugar concentrations, we can also conclude the same information, like the Km for sodium, dependent on the sugar. And as you can see, the Km in both cases increases if substrate co-substrate concentration decreases. So you have a cooperativity of substrate binding, which goes in both directions, concluding that you actually have a random binding order in place. So this, what I just showed you was only information, kinetic information about D-glucose, presence or absence of sodium. Now we also wanted to dig into sugar specificity. And what you can see here is measuring 7 different sugars in presence of sodium and in absence of sodium. It's replaced with potassium here. This was done here with 3 millimeter sensors, allowing for a higher signal to noise ratio. And for those sensors, the steady state actually dominates the signal. Let's have a look at the traces here in presence of sodium. The green curve is fructose, which is not a substrate for SGLT1. So there is no specific current here. This is basically in a negative control. And then you see the dark blue trace is the glucose, which is shown here. Again, you see a biphasic current decay. And then you also have 2 other substrates marked in blue, MDG and galactose, which look quite similar. And these 3 blue traces can be clearly distinguished from those red traces obtained for other sugar substrates. I call them minor substrates. For them, you actually see a right shift of the peak, which indicates that the pre-steady state component is much lower. And we confirmed that actually in absence of sodium, we really see that the pre-steady state current is lower. In contrast, the steady state actually is higher. So the peak of the slow current component is much higher compared to the major substrates here. Just as a comparison, we did the same experiments using 1 millimeter sensors, and it's quite interesting to see how the different sensors affect the currents you record. The basic results are the same, and the only difference is that for the 1 millimeter sensors, you have a better temporal resolution, so you can better fit the kinetics of the current decay. And also the pre-steady state component, if it's limited by the time resolution, will affect more the peak current you are measuring. So these currents here is just basically saturating concentrations of sugar. We then repeated the concentration sequences for all those sugars in presence and absence of sodium to actually conclude something about sugar specificity and the reason behind that specificity. First, we can have a look at the Imax, which basically correlates to the Vmax of the transporter. You can see that Imax doesn't really has huge differences between the different sugars. And interestingly, you see that glucose, which is actually the main substrate of SGLT1, has the lowest transport capacity. So we conclude that Imax is not really a specificity factor for SGLT1. What else? We can actually compare the affinity for the different sugars in absence of sodium. This reflects the affinity of the sugar to the empty carrier. And here you can see that glucose actually has the highest affinity, but still affinity is not very much different compared to the other sugar substrates. So it's not really a huge specificity factor again. Now, the affinity in presence of sodium, say you can actually see that the affinity is highly increased compared to the affinity to the empty carrier. Plus, this is only the case for the substrates which are before labeled in blue, the major substrates, glucose, MDG, and galactose, but not for OMG and xylose, where the affinity is essentially the same. So this tells us that cooperativity actually is a specificity factor. When sodium binds, specificity for sugar, for D-glucose, is enhanced. And now looking at the Km, which is basically the apparent affinity of the transport, we conclude that again from the transport current phase. And here you can see again that for the major substrates, the apparent affinity is much higher compared to the real affinity, which was concluded from the pre-steady state component. But again, this is only the case for the major substrates, not really for the OMG and xylose. So this means that the Km is the real specificity factor here in place, which really changes 160-fold across these different sugar substrates. This was it about different sugar species. We also had a look at different ions, ion selectivity. Here you see sodium concentration dependence. We performed different MDG concentration jumps here, always with 250 millimolar MDG, and we had different sodium concentrations. And you can see that you again can measure EC50 values for the pre-steady state component and for the transport component, reflecting the EC50 for sodium. Interestingly, if you apply a sodium gradient, then you will increase the transport component, which nicely fits to what has been published before. In literature, it's described that the sodium release step actually is the rate limiting step in sodium coupled sugar transport. And if you remove sodium from the internal solution, then you will basically speed up that rate limiting step, and you will have an increased transport current component. We did the same concentration sequence, replacing sodium with lithium. With lithium, you can see that there is not really a transport current phase resolved. So this means that the transport is below the resolution of the instrument, likely at least 10 times lower in transport rate compared to the sodium coupled transport. And we also did the gradient with lithium. And here you can see the transport current phase is not increased if you apply a gradient. And this means that lithium release is not the rate limiting step when you see lithium coupled transport, but it's rather likely lithium binding or the alternating excess of the carrier, because lithium probably gets or induces a transporter state, which not really favors the conformational transition, which is following and leading to transport. We also did proton-coupled transport. This was done by just replacing sodium or lithium with potassium. Potassium does not interact with SGLT1. So we can basically just measure different pHs to adjust or apply different proton concentrations. And as you can see, we actually see the transport current component increases with acidification. So we can conclude pK values from that fits, and also pK values for the pre-steady state current component. But similar as for lithium, if you have proton coupled transport, a pH gradient will not increase the transport current phase, meaning proton release is not the rate limiting step. Here, it's again likely proton binding or the following conformational transition. So what do we actually know about the pre-steady state current component? We don't know much about it. It's likely, I mentioned that before, an induced fit or maybe an occlusion step of the sugar molecule. But what we can investigate is how the charge translocation is affected by substrates. And here you can see there is the Qmax, which is basically the maximum integral we measured under saturating sugar concentration in presence and absence of sodium. And you can see that the Qmax is very similar for all the sugar species, no matter if you have sodium or if you don't have sodium. This means that all the sugars likely induce the same conformational, the same electrogenic conformation transition in SGLT, leading to a similar conformational state. And you basically see the same transition of charges across the membrane up on binding of different sugars. So what about the ions then? The sugar obviously doesn't affect the conformational state upon sugar binding, but we could find that the pre-steady state current is highly affected when you actually bind the sugar to the sodium bound carrier or the lithium bound or the proton bound carrier, as you can see here. Also, chloride has a huge effect. If we replace chloride by gluconate, also the pre-steady state component is massively decreased. So since sodium, lithium, and protons are all monovalent cations, they don't really directly contribute to the charge translocation. It's rather the state of SGLT1 up on sugar binding, which differs between those different bound states. There's actually another publication which was released half a year ago, where we claimed similar statements using the proton-coupled sugar transporter XyLe. And here, actually, different sugar species leads to different pre-steady state currents, and these really correlated with different conformational states we figured out from different structures available in the PDB database. So finally, we concluded an 11-state model, and we also applied rate constants according to what we've measured in those different surfer assays, and simulated this. And this actually simulates most of the kinetic data we obtained very well. We used random binding order. So we basically start here at state 1, which is the empty carrier, outward facing. And this carrier can either bind the sugar first, following that electrogenic conformation transition, then sodium, followed by another electrogenic conformation transition, and then alternating access to the inward facing side, substrate release, and back to the outward facing state. Alternatively, sodium can bind first. This is actually the commonly published ODAT binding model, which many people refer to. After sodium binding, we cannot see any electrogenic step, as I mentioned in the very first slide, but then the sugar binding generates a huge electrogenic transition, which basically is the double of electrogenicity compared to those two steps, which also matches with that model. With that model, we assume two rate limiting steps, which are the alternating access steps you can see here, which also explains our findings that Vmax actually also varies with the sodium concentration, with the internal sodium concentration, and with the substrate species. So to sum it up, we actually got a lot of kinetic data on SGLT1, and all these things can be applied to other transporters. Of course, we were lucky since SGLT1 actually has a pre-steady state current, which can be recorded and which provides additional information besides the actual transport current. Don't need to go into that anymore, but we basically measured lots of different kinetic parameters. From those, we concluded that 11-state kinetic model, which describes in some simulations all the currents we've recorded. These pre-steady state currents are triggered by sugar binding. That's an important difference to the pre-steady state currents which have been published up on voltage steps, which are related to the empty carrier. And we found that the ions actually define the conformational state up on binding of the sugar. And it might be the case that these electrogenic transitions are actually conserved across all kinds of sugar transporters. It is present in lots of different coupled bacterial sugar transporters, sodium-coupled ones, proton-coupled ones, and even those who perform unipotent modes. It's present in SGLTs, and maybe it's also present in some other human sugar transporters. So with that, I want to finish the part with SGLT1. I want to thank the whole Surfer team. Everybody kind of contributed to that. And especially to all the students who were involved, who spent their time during master thesis and bachelor thesis, and also Rocco, who was involved in kind of few of measurements. Yeah, and of course, also thank you for the funding, and Nana-Ion, who of course also supports all these internal projects. And yeah, with that, I hand over to Rocco, who will continue on his PhD results on the sodium-coupled GABA transporter.
Okay, thank you, Andra. So, hello everyone. I will quickly introduce myself. I am Rocco Zerlotti, and I am a PhD student here at Nana-Ion for the European Training Network, Neurotrans. My academic affiliation is with the Regensburg University, and I am a biomolecular chemist by training. So, with that, I would like to introduce you to my research project, which is the functional characterization of the human GAT1 through the solid supported membrane electrophysiology. Okay, so first thing first, a quick introduction about GAT1. So GAT1 is the main transporter in the central nervous system of the gamma-aminobutyric acid, which is the main inhibitory neurotransmitter in our body. So its main function is the reuptake of GABA from the synaptic cleft through a sodium and chloride dependent transport cycle, and it exploits the energy that comes from the sodium gradient that is maintained by the sodium-potassium pump. So the currently accepted stoichiometry is 2 sodium for each, 1 chloride and 1 GABA, but there is a debate on this. So some people think that for each GABA molecule, 3 sodium ions are actually co-transported. Anyway, the transport is electrogenic, so we can study it by means of electrophysiology. So here, Andra already gave an exhaustive introduction, so I just want to stress out some important points. So the current recordings are at 0 millivolt membrane voltage. All the currents are transient. So here you can see in blue the traces that we see on the SSM, and in red are the traces that can be seen on a, let's say, voltage clamp setup. So if we are in presence of a monophasic decaying slow transient, this basically identifies, or usually it identifies a stationary transport current, also known as a steady state. So basically a flat line that you can see here. We can also observe a fast transient monophasic decay. In this case, we are speaking about pre-steady state currents, and we see the same in a voltage clamp environment. So the pre-steady state is actually an out of equilibrium state. So these are very fast charge translocation across the membrane, and usually they are due to ion binding or substrate binding or conformational rearrangement of the transporter. And we can also have more complicated situation like this scenario in which we have a biphasic behavior of the current, like we have seen with the SGLT1. So we have a pre-steady state component and a steady state component, and this is what you may see in a voltage clamp environment. So just some terminology. So I will speak about EC50 and Km. So the EC50 is the substrate concentration to get half the maximum parameter value. This parameter can be the peak, can be the charge, or can be the current at a defined time point or whatsoever. Km is a particular case of EC50 and is obtained by estimating the concentration to get half the maximum transport current amplitude, which is proportional to half the maximum speed of the transport reaction. And it coincides with the Michaelis-Menten, the well-known Michaelis-Menten constant. Okay, so we can dive into it. First of all, the sample. So our sample is composed by membrane vesicles from CHO cells that are overexpressing our target GAT1. And the first thing that we wanted to do was, of course, validate the sample. So in order to do this, we worked in presence of a high sodium gradient. So we have no sodium inside the vesicle. Then we establish the sodium gradient by jumping with sodium chloride, 150 millimolar. And then we jump with GABA, so the transport reaction take place, and we see the electrogenic event. So as you can see from the traces here, we did the same thing on both CHO membrane vesicles expressing GAT1 and also empty CHO vesicles as control. And you can notice that the vesicle expressing GAT1 upon GABA jump trigger this very nice nanoampere amplitude peak, while empty CHO cells didn't trigger any significant current. Then to further validate the sample, we use the SKF, which is a very potent selective competitive inhibitor for GAT1. And you can see that the transport current actually decreases proportionally to the SKF concentration applied to all patterns. So here it is important to tell to speak about the shape of the GABA-induced current in SSM. So if we work in presence of a very low sodium gradient, so what we do is actually preloading the vesicle with a lot of sodium chloride, 135 millimolar. Then we establish this small gradient, jumping with 150 millimolar sodium chloride. So we have a 15 millimolar difference only between the outside and the inside. And then we activate the transport with a GABA jump. We notice that we obtain the current that you can see here. So this is very, very similar to the SGLT currents that Andrea showed before. So this is biphasic. We have this pre-steady state component and this slow decay steady state component. And in this situation, when these two phases are really distinguishable, we can use the algorithm that Andrea showed you before to reconstruct the current and obtain the transport current as it may be seen in a voltage clamp environment, which also gives us a better readout of the transport current itself. When we work in a very high sodium gradient, so no sodium inside, and then we jump with high sodium and then we GABA to trigger the transport, you can notice that the biphasic behavior is not really that distinguishable anymore. So in this case, the algorithm fails to reconstruct the current, but we can always measure the transport current by simply reading out the current at a specific time point. So now we may ask ourselves what kind of information we can grasp from this GABA-induced current. Okay, so if we are looking at the transport current, of course, we get nice kinetic information regarding the dynamic equilibrium of the full motion of the transporter. So basically, we can obtain the Km and Vmax. But we can also observe what happens with the peak, with the pre-steady state current. So in this case, we can study the peak amplitude and the decay time. So the peak amplitude, we notice that it is very nicely dependent on GABA concentration, and you can see from those response curves. And there is a big difference if we are measuring them in presence of a very low sodium gradient or in the presence of a very high sodium gradient. And as you can see, there is basically a 5-fold difference between the EC50 of the peaks in these two different conditions. Regarding the decay time, it is better to explain a little bit what we think that this pre-steady state current is. So here is a portion of the transport cycle for the GABA transporter. Here you can see the binding of GABA, and here you can see the successive step. So we asked ourselves, is this pre-steady state current like directly the GABA binding? This is very unlikely because GABA in physiological condition is baryonic, so it has no net charge. So it is really not likely that what we are seeing is the binding of GABA itself. What we may see, and this has also been postulated by these authors here, is that the electrogenic step actually is a successive step. So this step may be either the flipping of the carrier, the carrier is full and it flips in the inward facing conformation, or it can be the occlusion of the carrier, which I here like marked with this star. And in either case, if we study the decay time and more specifically the KOPS, so the reciprocal of the decay times, we can plot the KOPS points as a function of GABA concentration, and we can fit them to this model that we have seen before with Andre's presentation. And in this case, we can obtain information about the KD, so the affinity for GABA, or the thermodynamic affinity for GABA of the transporter, and also the rate constant of this successive step, being this the flipping of the carrier or the occlusion. So moving on, we notice that the sodium gradient has a very big effect on the GABA-induced currents, so we wanted to further investigate it. So we measured the Km of GABA in low sodium gradient and in very high sodium gradient. And surprisingly, actually the Km didn't change too much between these conditions. So GABA Km is not really affected by sodium gradient. And this also was nice because the Km we measured is really close to the literature data reported at very small voltages on xenobus sulfides with 2 electron voltage gram. What changes in sodium gradient actually is either the Vmax of the transporter, so the maximum transport current, and the pre-steady state or the peak amplitude. So here the schematic of, very quickly, the schematic of the experiment. So we have different internal sodium preloading concentration. We jump with our 150 millimolar sodium, and then we trigger with supersaturating GABA. And here I report the data. So this is the parameter, either the peak amplitude or the transport current, normalized by normalized to the maximum sodium gradient that we can obtain. So 100, no sodium inside and 150 millimolar sodium jump, as a function of decreasing sodium gradient. And as you can see, both the transport current amplitude and the peak amplitude nicely decrease with decreasing the sodium gradient. So those two parameters are affected. So we also wanted to study the external sodium concentration, the effect of external sodium concentration. So in this case, we have no sodium inside. We just establish the gradient with different sodium concentration, and for each sodium concentration, we measure a dose response curve. So here I present the data as this 3D plot. So we have here the current that is normalized to the maximum GABA concentration used for each sodium concentration as a function of both sodium and GABA concentration. So here you can observe basically a wireframe that is composed by those response curves, either GABA versus sodium or sodium versus GABA. Here on your right, you can see how GABA Km is affected by external sodium concentration. And in this case, GABA Km is very highly affected by external sodium concentration. If we go very low on sodium, so we have 50 millimolar sodium concentration outside, GABA Km reaches the millimolar range. So we also asked ourselves if it was possible to measure pre-steady state currents, so electrogenic events, upon just ion binding. So in absence of GABA, we just jumped with sodium and chloride. So regarding sodium, here we have our vesicle without, of course, sodium inside, and we jump with sodium, and this triggers an electrogenic event. We have to be careful here because in SSM, when you do ion jumps, you always you trigger artifact. I mean, you always have an electrogenic event, but this may be only an artifact. So what we do is repeat the same experiment with also empty CHO vesicles as control. And what then we do is just make the difference between the GAT1 current and the empty CHO current. So we did this with counteranion chloride and gluconate to see if some difference may arise. So when we use chloride, it was possible to observe net, albeit small, net currents for sodium binding, which and we measured also the EC50 of the peaks here, and we measured this 13 millimolar EC50. Very interestingly, when we switched to gluconate, it was impossible to observe any net current. Now, this is very interesting because it has been widely postulated that chloride, the role of chloride in GAT1 is that of stabilizing sodium binding. And in this case, this may be an evidence for that, because when we are in presence of chloride, we can see currents that are more or less directly caused by sodium binding, while when we jump with gluconate, we don't see anything. So we did the same with chloride, so chloride jumps, and in this case, we used as countercation, we used both potassium and sodium. In this case, it was really no different using potassium or using sodium. We obtained these nice negative currents, which also follow with a very similar kinetic. So we have a peak EC50 of around 100 millimolar. And this tells us something about the order of binding of the ions, because of course, at this point, we may say that if chloride is present, then chloride should bind first. Then sodium can bind and is stabilized by chloride. Then GABA binds, further stabilizes the sodium and all the protein conformation, and then the transform may happen. So we proceeded also by studying the reverse transport current. So basically GABA efflux instead of GABA intake. To do that, we preloaded the vesicle with the sodium and GABA. And then basically what we did was to establish outward directed gradients. So we first we remove sodium from the outside, and then we remove GABA, and this was able to trigger the reverse transport. Here you can see the traces as example, and what we found is that the reverse transport and the forward transport seem to be very asymmetric. If you focus here on the table, I report the Km for the forward transport for GABA and sodium, and for always for GABA and sodium, but for reverse transport. If you notice, we have basically a 200-fold increase of Km in the reverse transport mode. So basically, that says us that we need a big load of GABA to trigger the reverse transport. While for sodium is the other way around. So for sodium, we need less sodium inside, 3-fold less to be precise, to trigger the reverse transport with respect to the forward transport mode. So what this tells us, I want you to focus here on the scheme on your left. This is like before. This is a portion of the transport cycle for of GAT1. Here I report the inward-facing sodium-bound conformation of GAT1. Here we have the sodium release inside, and then we have the empty inward-facing human GAT1 and the flipping towards the outward-facing state. So here the transport cycle can start again. At this point, what happens if we have this higher affinity for sodium inside, and we are in a situation in which we have a very high local sodium concentration, like for example during the neurofiring event, then this may result in an impaired capability of releasing sodium in the inward-facing state. So basically here the transporter is locked in the inward sodium-bound facing state, and so less transporter is available to restart the cycle and transport GABA. So some evidence for this comes from observing the pre-steady state current that I showed you before. So when we are in absence of sodium inside, we have the EC50 is in the micromolar range, and the peak amplitude is 5 nanoampere height. While when we are in presence of high sodium inside, we need more GABA to trigger the pre-steady state current, and also the peak itself is very smaller. Also the Vmax, so the maximum transport current, it is very different. So if we are in presence of sodium inside, high sodium inside, we have the Vmax is only 20% of the Vmax that we have when no sodium inside is present. So to finish, we also wanted to have a look at the stoichiometry. So here on your left is the physical principle for the essay. I won't go into the detail, but I just want to tell that we exploit the fact that we are at zero membrane voltage, and so basically the delta G is only a function of chemical potential. And at this point, when the delta G is 0, which in this graph here on your right corresponds to a 0 charge, then we have that the ratio of the chemical potential that here is reported on the x-axis corresponds to the stoichiometry for the substrate that we took into account. So we took into account the sodium and GABA. So we preloaded the vesicle with a certain amount of sodium and a certain amount of GABA with the symmetric condition outside, and then we operate a double jump here. So jumping with different sodium and GABA concentration at the same time. Of course, we had the negative controls. We are jumping with with sodium, so ion jump, and we get artifacts. And we were able to assess that at 0 millivolt, the stoichiometry for sodium and GABA seems to be 2 to 1. Okay, so just to sum it up, the take-home messages are that the GABA-induced currents in solid supported membrane are composed by this fast pre-steady state component and the transport current, and that this pre-steady state is likely to be related to a conformational change on the human GAT1 after GABA binding. then the sodium external concentration can affect both the transport rate or the speed and the GABA Km, while the sodium gradient seems to affect the GABA speed, the transporter speed, but not the GABA Km. Both sodium and chloride alone can generate fast steady state current upon binding in absence of GABA, and since sodium cannot generate this current in absence of chloride, chloride probably is necessary to stabilize sodium binding. That the human GAT1 transport is highly asymmetric. So the efflux, the GABA Km in efflux is 200-fold higher with respect to the forward transport. So we have a possible lower affinity for GABA in the inward facing conformation, while the opposite happens for sodium. So we can have a possible higher affinity for sodium in the inward facing conformation. And at the end, that on SSM, the stoichiometer GABA sodium appears to be confirmed to be 1 to 2. So with that, I am finished, and I thank you for listening.
Thanks, Rocco and Andre, for going through the presentation. Just want to welcome everyone in the audience to submit your questions. I'll go ahead now. I'm mindful that we have a couple minutes left before the top of the hour, so please do submit your questions if you do have some in the chat, and we're happy to address them to Rocco and to Andre. Andre, I have a question for you. To analyze your pre-steady state currents, you have used the decay of the current. What about the rise time? Does it also contain kinetic information about the transporter?
Maybe. It mainly depends on how fast the kinetics of the transporter is. So I think the rise time reflects the time from the initial state of the transporter to the first electrogenic step, which takes place in the reaction cycle. If that is very early, if the electrogenic step is very early in the reaction cycle, then the rise time might be limited just by the time resolution of the instrument, which I think is the case in many situations for many transporters. So yeah, it might contain some meaningful rate constants, but not in all cases.
And Rocco, question for you. Have you ever checked the orientation of the protein in the membrane?
So no, I didn't. There are quite a few possibilities for that. So we can use either specific antibodies, or we can also use a combination of selective inhibitors, for example, inhibitors that act only on one side of the membrane for that one. But I do think anyway that we have a preferential orientation for the protein, since I see that the transport is highly asymmetric. If we have no preferential orientation, I would say that probably we wouldn't see any difference in the two transport modes.
Question for you again, Andre. Your peak current sugar dependency indicates maybe a substrate inhibition. Is that because the transporter is not saturated with sodium, or release of sugar is inhibited?
I'm not sure if I understand the question. Can you repeat it once more? The peak current indicates inhibition?
So your peak current sugar dependency indicates, in parentheses, maybe a substrate. It's in relation to one of your first slides. A substrate inhibition, is that because the transporter is not saturated with sodium, or it releases sugar is inhibited?
I got it now. So yes, the peak, the pre-steady state peak is reduced in absence of sodium. This in the first slide, yes, this was due to the electrogenicity is actually lower in absence of sodium. As you might remember, in the 11-state model, we have if the sugar binds first, we have two electrogenic steps, which is the sugar binding, or the reaction which is following the sugar binding, and then also the reaction which is following the sodium binding. Both of these reactions together have the same electrogenicity as the sugar binding to the sodium bound carrier. So at the end, it matches, it fits together. It's just that basically, if the sugar binding happens to the empty carrier, that's not a complete transition what is happening. You need the sodium binding afterwards to have a complete transition, which transfers the complete charge in the whole cycle, basically.
And then back to you, Rocco, question for you. Did you ever try using valinomycin to generate a negative membrane potential?
Yes, I tried that once. I plan to try it again, actually, and get more quantitative results. And I observed an effect on the current. So basically, the peak is almost double when we use valinomycin. And so we have a negative membrane potential. This is also a good indication that the sample is good, actually, because as we know, I mean, GAT1 is voltage dependent in its transport reaction. So the fact that the vesicles are responding to a negative potential is a good indicator that the sample is good.
And then a final question for you, Andre. If one wants to be on the safe side, what control experiments can one perform to distinguish transport from pre-steady state currents on the surfer?
So if you observe a monophasic decay, then you can't be really sure about if it's transport or pre-steady state. If you observe something biphasic, as for SGLT1, then of course, at least one of those currents needs to be pre-steady state. But for monophasic current, the only thing you can actually do is trying to get samples with different lipid to protein ratios, and then see if the decay time of your current actually changes with the protein density inside the sample. This is because if you have more protein inside your sample, you will charge the membrane faster when you trigger transport, because there's a higher total transport capacity. And this faster charging will lead to a faster decay of your transport current phase. But pre-steady state current decays are basically an inherent property of the reaction you are measuring, and these won't be affected by just changing the protein density. And that's a way how you can distinguish. Of course, this is more easily done if you have protoliposomes measured, then you can adjust it. For membrane vesicles, it's more difficult to try something like that.
Great, thanks, Andre and Rocco, again for that great presentation. For everyone in the audience, we will make this available on demand. For those of you that were not able to join throughout the course of the presentation, you could rewatch it. I see that we have one member that, Laura, if you have, I think you're raising your hand. If you want to ask a question, I also welcome you if you would like to do so like that. Otherwise, I would close out the session for today. And I'm sure that Andre and Rocco are both happy to receive your questions.